a talk really about how not to do science, and just to teach you some of the problems that turn up, and to teach you a little bit of criticism about genetic findings. And to do so, I'm going to take my examples from the genetic basis of personality. So I'm going to start by explaining to you how we define personality as psychologists, and then I'll take you through just a very tiny bit of genetics. You don't need to know very much, and then I'll show you the examples and let you make your own conclusions. So let's just start with personality. So most people think they know what personality is, where we spend our lives judging other people's personality. We're trying to work out whether you're someone I want to take out for a meal, and I might do so and realize at the end of the meal you're not so nice. And you also realize that some people get married and after 10 years realize that they're living with a psychopath. And other people realize that it's quite hard to make these decisions. Now, in personality psychology, we use things like this. These are personality questionnaires. They're very popular. People like to do this. And it's curious because on one hand we know that judging personality is hard, but on the other hand we trust, or we like to think we trust, the results of these types of surveys. And what's interesting is that if you read the instructions here, it says, please answer each question by putting a circle around the yes or the no following the question. There are no right or wrong answers, no trick questions, work quickly, and do not think too long about the exact meaning of the question. Well, of course, most people take a very opposite attitude to this. And firstly, most people think the answers that are given are really important. You've got to get it precise, and they spend quite a long time worrying about it. They also think that there might be trick questions. And actually, I should tell you that, so this is the ISENC personality questionnaire, and don't tell anyone I told you this, but there are some trick questions. So if you look at this, you'll see question number 4, have you ever taken the praise for something you knew someone else had really done? So if you say, no, I've never taken the praise for something else, then you'd score 1 on ISENC's lie scale, because he believed that people shouldn't say things like that. But that's actually not mostly, in fact, that's not really how anyone now uses that scale, and there's other information here that we're interested in, but let me just emphasize this with a bit more information for you. So here's the second page of the questionnaire, and I want to draw your attention to question number 61. So you can read just down here. It says, is or was your mother a good woman? And you can see that the person who's answered this, they've filled it up correctly, just say yes or no answer. But you see, some people don't do that, and when we send these questionnaires out, sometimes somebody writes back to us. So question 61, I found this very hard to answer. My mother was good in that she provided us with clean clothes, food, tidy and clean house, but she was and is emotionally cold and lacks empathy. I can understand why this is, but since warmth and empathy is so essential to the well-being of children, I find it hard to deal with her, as over time her cruelty seems to have increased. She had a hard childhood and years of abuse from my father. It's not surprising, but it's hard to say whether she's good or not. Okay, so it's a nice considered answer, but it's not a yes or no answer. So this woman has not really understood what we're trying to do here, which is to answer the questions quickly, not think too much about them, and just give us a yes or no answer. So that's not how you should think about the questionnaires. So you might be saying to yourself, well, how should I think about them? What does it mean if we say yes or no? So let me just give you a couple of examples. So I've just taken 2 questions from the ISENC personality questionnaire. Are your feelings hurt and do ideas run through your head so you cannot sleep? So logically, there is no connection. There's no reason why, if you say your feelings are easily hurt, you should then later say, yes, ideas run through my head so I cannot sleep. But when we give these questions out to large groups of people, we will find that there is a correlation. So people who answer yes to one are more likely to say yes to the other. And indeed, the questions are so chosen that they do form a pattern of correlation. Not exact, we don't want to ask the same question twice, but there is a degree of correlation. And what the personality psychologists do is that they take that pattern of correlation, and from that they derive a factor. And the ones that we're most interested in at the moment is one called neuroticism. So neuroticism is defined as a pattern of correlation across those answers. And it's interesting because although it doesn't really matter what you've said, whether you believe the actual answers or not, if you score highly on the N scale, then it's a strong predictor of the onset of depression. So we're defining neuroticism by the pattern of correlations, and then we use it in another sample, maybe the same sample, to look at other features, and one of which is depression. And we can also say that it's subject to genetic effects that are shared with depression. So in other words, this is a personality measure which indicates whether you might be depressed and also shares some of the same genetic factors. I'll tell you a little bit more. So if you just look at these questions, you can probably see the questions that are picking up neuroticism. Are your feelings easily hurt? Do you often feel fed up? Are you often troubled by feelings of guilt? Those typically pick up the sorts of problems the sorts of features that the psychologists are interested in. So although I've said it's not entirely, we don't really care whether you say how much you think about these answers, but actually you can see these are picking up some change in mood. So that's all I want to tell you now about personality, and we'll talk very briefly about genetics. So I'm sure you all know that you get one copy of DNA from your mum, one from your dad, and that there are variants in there, so that I've shown here in red the positions in the genome where you'll find places they differ. Geneticists refer to those as alleles, and because you've got two alleles, you can form three genotypes, two Gs, the heterozygote and the homozygote for the other class. And in the types of work we're looking at, all that happens is that people ask the question, is there a difference in the personality measure across those three genotypes? So it might be that you're severely neurotic, moderately neurotic, or unaffected, and not neurotic if you have that CC. So that's really all the genetic analysis that's required to understand what we're going on. And this is something that's been done an awful lot in psychiatry. I just put into PubMed terms looking for the numbers of publications using that association approach. This is the number of publications, and these are the years. So I did this last year, 2016. And you can see that by about the year, this was discovered sometime about the late 1980s as an approach. And about the year 2000, there was about one publication a day coming out, over 365. We're now up to almost, getting on for 10 a day. I mean, it's really a very productive field. So lots of people are doing this in psychiatric genetics. So if that's so, what we'd like to know is how much have we learnt? What type of information is it producing us? So let me just give you a couple of examples now. So now you need to know a little bit of neurobiology, very little bit. So I've told you about neuroticism, that's the personality phenotype. And I've told you what we mean by association. So now we need to look a bit at the molecular biology. And there's a gene, it's called the serotonin transporter, which I've written up there. Serotonin transporter is the target for Prozac. It's the one of the major drugs used for treating depression. It does so by blocking this transporter, and by so doing increasing the amount of serotonin that's available in a synapse. Now, there is a polymorphism in this gene. So the gene is indicated here. It's its first exon, there'll be a second exon, and so down here. And just to the front of the gene, there's a control region controlling the amount of gene that's produced. And this control region contains a bit of DNA that's repeated. And in the middle of that repeat, there's either an insertion or deletion. So if you regard that just like I showed you the previous figure where we have two alleles, the two alleles in this case are going to be either the deletion or the insertion. And because we've got two alleles, we can obviously represent them like this as a short and a long, and then we can generate three genotypes, which I'm showing like this. So then the association test is simply to ask if there's differences in the scores, in this case of neuroticism, across those three genotypes. So for example, These are the numbers of the genotypes, and these are the scores. And if you carry out a test to see whether that's significantly different, it's less than 0.05. And this was done in 1996, and this is the paper that reported that finding, and it's now generated more than 3,250 citations. And to put that into perspective, if you get to be a scientist and if you publish a paper with more than 100 citations, you're doing really well. Anything over 1,000 is really remarkably high, and to get 3,000 is just extraordinary. So that would indicate that this is an important finding. And now we're going to see whether that's true. So after that paper came out, other people tried to do the same thing. It's quite easy to do, measure personality. It's quite easy to do that genetic analysis. And this was just taking the number of subjects in the study, the date, and who did the study. This is the p-value, and some people find it's less than 0.05, some people find it's not so, and clearly you'd like to know who's correct. So there are sort of two approaches to answering that question. And this is my friend, Marcus Manufo, and he and I started this work in the mid-1990s, asking the question, could we use what's called a meta-analysis to decide whether the studies were giving a consistent or not answer. And in a meta-analysis, you take all the published studies and you summarize them in a form like this. That's the name of the study here. And what we do is produce something called a standardized mean difference between the genotypes. That's SMD at the top there. And the difference is indicated by this little tick here, and the horizontal bar represents the confidence by which that mean is estimated. And then when you've put it in this form, you can add everything up. And when you add everything up, you get a total down here. And what you want is for these bars not to overlap 0. If the bar doesn't overlap 0, as you can see here, that means it's significant. If it overlaps 0, then it's not significant. So we want to see whether that's not significant. And in fact, if you look here, it says total, the confidence interval. And actually, it doesn't overlap 0. So it's the test for the effect here is given. So it looks like it is significant. But there's one very important proviso when you do these studies. And that's that you want them all to be testing more or less the same thing. So for example, you want them to be maybe testing men and women, not just one sex. You want them to be using the same questionnaire, or at least something that's pretty similar. And in order to know whether that's true, we want to see whether this distribution is drawn from something that's pretty homogeneous. So we have a test of heterogeneity, and that's given down here. The test of heterogeneity is actually significant. It's less than 0.05. So there's some evidence that people are testing not quite the same thing, so we want to correct for that. So we put in a correction for that, and then when we do, we can run the meta-analysis, and then we can find that the evidence for the association between N, that's neuroticism, and the serotonin transporter is the following. So we published that result and thought to ourselves, that's the end of the story, there's no effect, people wouldn't be interested in it. But there were some arguments. People said it's only a meta-analysis and you've got this heterogeneity problem, so we don't really think you've really nailed this. So the alternative to doing a meta-analysis is to carry out a much bigger, better powered study. So we did that, and we carried out 2, in fact, with colleagues in the UK. And the way we designed these studies was as follows. We said, look, the distribution of the personality scale is pretty normal in the population. It's very easy to collect this information. I can send you out a questionnaire for you to fill in, so I can collect lots of data quite quickly. And the interesting part for me is to look at the tails of the distribution. So the genetic effect is mostly concentrated in the tails of the distribution. If I just get data from this group and analyze it, I'll get about 80%. And that saves me having to deal with the vast amount of information, the vast amount of data in the middle. So we sent off this questionnaire, it's the one I showed you before, and we sent it off to about, in the end, almost 200,000 people. We had a nicely big powered study. So this is when I was in Oxford, which is up here, and we sent this questionnaire out to this whole region there, and everyone sends back a questionnaire. And we got in the end 88,000 we got back from that in the end. And then we make a nice plot to show that the data look good. And so you'll remember what I'm going to do is ask this group and this group to send us a sample of, in this case, a cheek swab so we can extract DNA. Now, when you write a grant later in your life, if you become a scientist, you would write statements as I've just put them. But what you might not have thought about is, let's say, the social context in which this happens. So just consider this for a moment. So what we've done is we've sent out questionnaires to a large number of people, and then we're going to select the most neurotic people, the most highly neurotic people in all of the west of England. These guys are laid back and calm. They don't really care about this stuff, but these guys are the most highly neurotic people. So we're going to ask them to send us a cheek swab. So what's going to happen? So that's the cutoff point, 2 or 3 standard deviations, and then we send off to the neurotic family and ask them for a swab request. And of course, some people write back and tell us things. I've just received your request to send a mouth swab. This is the last straw. My holiday has been cancelled. My son is influenza. My husband has left me. And now this from you. It is the end. So quite clearly, we are getting the most neurotic people. So this strategy works in identifying people, but it has the consequence that they're not all going to want to take part because they're very, very neurotic. So when they write back, not all of them give us what we want. So we get an ascertainment bias because a group of people are not going to want to take part. That said, we've got a highly selected sample from the extremes with a lot of power to detect an effect. So now we're going to look at our results. So these are the genotypes. Remember, there's an L and an S. Those are the 3 genotypes here. And this is the neuroticism score. So the first thing to note is the big differences between these 2 bars. That's because we're assaying men and women. And men are not as neurotic as women. Okay? That's what this comes out every single time we do this. Robust result. But we're not interested in that. What we're interested in is the difference this way. So is this bar bigger than this bar or bigger than that one? No. You don't need to run a statistical test. There's no difference in the size of that. No effect at all. And just to be absolutely certain, I collaborated with my friends who live out in Cambridge, and they'd run a similar study on the east of England, and they sent out to 21,000 people, and then they selected 5,000 of those for genotyping, just using the same strategy. Oh, and they got exactly the same result as we did. So we thought that was basically the end of it. There's no effect of that polymorphism on neuroticism. But while we're doing all of that work, other groups around the world are doing things. And another paper is published. So this also is published in the journal called Science. And this paper is called Influence of Life Stress on Depression, Moderation by Polymorphism in the 5-HTT gene. So let's go through this. So influence of life stress on depression. So in fact, as you'll see in a moment, it's not really depression, it's risk of depression. And you now know that personality is a risk factor for depression, so it's really just personality. 5-HTT gene, that's the serotonin transporter gene again. And it's different because unlike what you've seen before, we're talking about influence of life stress on depression. So there's another term in here we'll look at. Notice that this has got even more citations, even more. One of the most highly, in fact, I think for some years, the most highly cited paper in all of neuroscience. And neuroscience is a much bigger discipline than genetics, so this is really very influential paper. And in brief, what these people argued was the reason that I couldn't see anything in the Serotonin Transporter Association is because we hadn't taken into account the environment. Let me show you what they mean. So there's a very famous figure from that paper. This is the figure. Let's go through the axes first. So the probability of major depression, that's the neuroticism score, that's personality. If you've got a high score, you're more likely to get depressed. So that's that axis. This axis, let's just deal with these first. So those are the 3 genotypes. Familiar with that, SS, SL, and LL. And when there's a difference between them, there's a genetic effect. And when there's no difference between them, there's no genetic effect. So what's different about this graph from what you've seen before is obviously on the horizontal axis. And this refers to child abuse. Really bad child abuse, never let you out to go and see your friends, and here sort of probably looked after you reasonably well. So this shows you that if bad things happened to you when you were a kid, if your parents really looked after you badly, then you would see a genetic effect that was mediated by the serotonin transporter gene. So in other words, the genetic effect depends upon the environment. So when I carried out the study I've just explained to you, we never asked our participants if they'd had a bad childhood or not, though some of them did tell us, of course. That we should have got, because these people argue the genetic effect depends on the environment. So that's why that paper was important. So that's not much more information to collect. Neuroticism score, genotype, and a question about whether you had a bad childhood. So many people start doing that, and guess what? People come up with inconsistent results. Some find it, some don't. But there's a little wrinkle here. When people said that they replicated those initial findings, they would mean we found a significant result, just like the other people had done. Is that really so? Now, that's the figure I showed you, and I'm going to try and simplify this. So I'm going to show here, this is the environment, and that's the risk of depression. And I'm going to show not 3 genotypes, but 2, just to make it easy. And what I'm showing at the top is that there's a genetic effect, because there's a difference between the 2 genotypes, but the lines are parallel. That's to say, the difference between the 2 genotypes is the same here as here, which means that there's no dependence upon the environment. The lines go up because there is an effect of the environment. If the environment's worse, you have a greater risk. Here, the lines splay out. So we've got one where there's no effect of the environment and one where there is an effect of the environment. But the fact I've drawn the lines like that, well, I could draw them like this. That's a bit different because they don't meet at 0 like the first figure. Or I could draw them like that, or indeed I could suggest there's some curve, or they could cross over in a different place. And each of those obviously has a different interpretation. But each of those could be significant. In each case, like this one here, there's a difference on the environment, so there's no effect here and a big effect here, but it's not the same. This will be significant, but it's not the same as the one I showed you before. But a p-value could be still significant. Now, here is a figure from a paper that claims that they replicated the original results. And you indeed appear to see something which shows that. So it has the genotype here, and it has the proportion of females that are going to get, this is the neuroticism score. And on the this axis, these are the high environmental risk and low environmental risk. So they've changed the axes. Rather than having the environmental risk here and the genotypes there, they swap them round. So it looks the same, but is it? So what we're going to do is plot it with the same axes as in the original paper. That's what it looks like. So then the question is that figure the same as that one? Well, no, it's not. The lines are crossing over. Here, they meet at the origin. There's a difference here. But this group just says, oh yes, it's a significant result, and therefore we'll say it's the same. So that makes it a little hard for us to compare all of these results. So with my friend Marcus Manufo, we carried out a meta-analysis. We take all of these data. We have to be careful about this problem, but we do that. And we found nothing. And at the same time as we were doing that, some colleagues, in fact Neil Risch, who's in UCSF, was doing the thing at the same time. We both found the same, that there was really nothing at all. And that led to a little article in Science saying that the psychiatric geneticists really didn't know what they were doing, and as a consequence of this, we got some hate mail, true hate mail. So we'll leave that story for the moment. I'll come back to it a bit later, but I want now to take a slightly different tack on this problem and introduce you to a story from neurobiology. But don't forget what you've just learned, it's important. So I'm going to tell you about an idea that's been around for about 10 or 15 years. And the idea is about something called intermediate phenotypes, or you'll hear the term endophenotypes. And the idea of the endophenotype is, I think, best shown in this figure. So I'm a psychiatrist. And psychiatrists work with things like depression and schizophrenia. So if you come to see me as my patient, we'll talk. We'll just talk. It's not a very scientific discipline. It's all grey and messy, just working out whether you're depressed or not. But I have colleagues who are psychologists, and they work with these things like emotional regulation, personality, episodic memory, reward. They're colourful, and they use things like questionnaires where you actually have measurements, so it's a little bit more scientific. And then there are people called neuroscientists who work with the brain. The brain has Latin names. It's a bit more scientific than what the psychologists are dealing with, and you can measure it. You can put brains into fMRI scanners, and you can cut them up and actually really measure stuff. But then there's another group of people, real scientists down the end here, molecular biologists, and they work with DNA. And DNA, it's got complicated names, and it's a molecule, so it's really hard science. And you'll notice there's an arrow running from here, getting sort of further to the right, and with a little arrowhead on the end. And what that suggests is that there's a movement in this direction, from the really hardcore science through the sort of pretty mushy neuroscience to the not so good psychology, and what the psychiatrists do, we don't really want to talk about. So that would imply it's a lot easier to be working with these sorts of phenotypes, which are closer to the really hard science, than the things that I work with. So the endophenotype captures that idea by saying you should be working with stuff over here rather than stuff over here, if you're going to understand genetic effects. So that's captured in this statement here. The assumption of the intermediate phenotype strategy is that genetic effects at the level of the brain are a more direct effect of genetic variation than is complex behavior. So that's just a simple summary of what I just told you. That implies that the genetic effects on the brain are larger, bigger, easier to detect, more direct. That's an easy hypothesis for us to test. Let's take a paper. My favourite magazine, Science. So, Serotonin Transporter, all happy with that. Genetic variation at it, you know about that. The response of the human amygdala, that's a small region of the brain that's involved in processing fear. Let's look at the experiment. So this is an imaging experiment. We put your head into put your brain into a scanner, and then we give you a fearful stimulus, and we look at the activation of the amygdala. That's shown by this little red area here. What we ask is there a difference in activation? And this experiment was done twice, first cohort, second cohort, and the difference we're asking is between genotype, and you look at the top, it says group, S group and L group, between the short allele carriers and the long allele carriers. So these are the genotypes here. In this study they lumped together SS and SL. We can argue about that later, but give them the benefit of the doubt. And there's a big difference here, and the p-value is significant at less than 0.05. Now, the claim is that this is a big effect, bigger than what we see in the complex behavior, bigger than for neuroticism. So let's just do something that the authors don't do and give us the effect size. So we're saying that the genetic effect size here is expressed as a percent, the percent of variation in that signal. So more than a quarter of the variation, 28%, is due to that single locus. That's the claim from that paper. Now you'll be familiar with what happens next. Again, it's not a difficult experiment to do, so lots of people start doing it, and then we can come and we can carry out a meta-analysis. I'm not going to bore you with showing you all of that results, but I'm going to ask a slightly different question, and that question is, how well powered are those studies? So we see there's problems here, but just what would we expect to see? So what we do for this is we say, okay, let's just assume that the studies are correct. They're picking up some effect size. Let's make the assumption that they find something, and let's work out their power to detect that. So the studies of amygdala activation indicate that an average sample size here was 32. It's a very small sample size for many studies, but they claim it's big enough. And we work out from that what the power should be to detect the effects they report. We don't know what the effect is, but we just assume they get it right. And very surprisingly, we find that the average power is about 15%. So again, later in your career, when you're writing a grant, never write that your power is 15% because you won't get the grant. You have to say it's well powered. That's 80 or more %. These are clearly not well powered. Okay, so that's our first conclusion. But we can sort of push this argument a bit further, and there's an epidemiologist, John Leonardis, who's done a lot of this work, and he has a number of papers. I'm just showing you one here, when he looked across all of the brain volume studies, and he reports here that there is an excess significance bias. In other words, if you have a low power, then you probably are reporting something which isn't really there. And he argues that in fact that literature is full of people who are reporting things which are actually probably not true. And he does this by working out the number of expected studies with statistically significant results is estimated, and then he compares that against the number of observed significant results in those studies. So again, the assumption here is that we don't really know what's going on, so we just take everyone at face value. They give us the effect size. We work out the effect size from everyone's studies, and then say, okay, you guys say overall that it's an effect size of 5 or 10%. How well is your study powered to detect that effect size? Just as I've shown you before. And his argument from this paper is in fact that there's an excess of significance. So we can take that even further. And in fact, a couple of years ago, we did this across almost all of neuroscience. We looked at imaging studies, animal behavior studies, genetic studies. We applied this approach, but it's relatively easy to do. Just pull out all of those meta-analysis. And indeed, we saw that there was power failure. So, in fact, small sample sizes are rife. People are working with power, as you'll see in the moment, which is just well below what they should be doing. So here is the key figure from that paper. This is the number of studies on this axis, and this is the power. So remember, what we should be having are power greater than 80%. Oh, that's this little group here. So 15% of all of those studies we would regard as well-powered. And all of these ones, and look, there's some down here at less than 10%. Like a lot of studies, really, really, really underpowered. But the majority of these studies are published, and they're published because they report a significant effect size. And as a general rule, if you report a significant effect size and you're underpowered, that's a false finding. So one interpretation, and I'll say this to you because you're not going to write it out and I'm not going to get into trouble for saying it, one interpretation is that the vast majority of the findings in neuroscience are wrong. They're just simply false positives. But we don't like to say that too much because I have colleagues in this building and they won't like me for saying those things. So let's just turn finally to look at another issue. So genome-wide association study is what I've shown you before, same approach. Instead of looking at one gene, one locus, one allele, one genotype, we look at the whole genome, and they're reported in studies that look like this. So again, there's a significance. Each of these numbers represents a chromosome, and each of these tiny little dots, the blue ones here, represents the result from a single analysis. And when they're green, then the investigators believe that they found a significant result. So this technology came along in about 2007. It's dominated the field since. And a number of people have applied it to the sorts of phenotypes that we're interested in. So let's just go through those. So here's the results for my friend Neuroticism, the personality type that I introduced you to at the beginning. And you can see that there are some significant findings here. And we're interested, of course, to know about the serotonin transporter, and that's on chromosome 17 here. Oh, there is an effect on chromosome 17, but no, it's not the serotonin transporter. That's right down here, nothing there at all. So that was a little bit disappointing. But now let's go to that other issue that we talked about. Remember the intermediate phenotype strategy. And what does that look like? So we don't have one for amygdala activation yet, but we do have something pretty similar, which is just looking at parts of the brain size. Remember, you can look at the brain and measure it. So what do those results look like? Oh yes, there's some effects there. So now we want to ask the question, are the effects we see from this study much bigger than the ones we see in this study? So we can look at those loci and just look at the effects of them. In fact, I'll do better than that. And here I've plotted out all studies for everything, absolutely everything. That's very useful because it gives us a ground for what we'd expect to see. So this is expressed as a percentage. And you can see that the median is down here at about 0.02 or 3%. Tiny, tiny effects. So the vast majority of these studies find very little. Now, remember, we said the intermediate phenotype strategy is expecting to find large effect sizes. And I worked out for you the effect size from that science paper, and that was at 28%. That will be out here. So the question is that actually true? No one in any genome-wide association study for any phenotype, nothing, absolutely nowhere, ever found anything at 28%. So that's another indication that things are maybe not quite what they should be in this field. So I want to spend the last few minutes just addressing that question. Why should it be? Why are so many people publishing things which are probably wrong? So we can approach this scientifically. This is something you can ask, and a question actually anyone in this room can answer, because the information you need to answer this lies in the publications themselves. You can pull out measures of what they've done, who they are, and use that to answer this question. So I'll give you a couple of clues as to how you might address this. So one thing is, could it be possible that where people work is important for the results they give? So we can answer that question by looking in the meta-analysis and seeing where people live determines their results. And we'll use a measure of effect size. This is an odds ratio, not a percentage this time, an odds ratio. An odds ratio of 1 means you don't see anything. If it's greater than 1, there's an effect. And you see that in North America, people tend to publish things with a positive effect size, whereas in other parts of the world, they don't. So that would indicate something interesting. But then you might ask, well, what is it about America? So we thought about this a little bit, and in order to answer that question, my friend Marcus and I came up with something called a bias index, where we divided the individual study, the odds ratio, that's the effect we're looking at, by the pooled odds ratio. That's the odds ratio we get from the meta-analysis. We divide what we are told in the study by the total, and that gives us an estimate of the degree to which any individual study over or underestimates the true effect size. That's true in inverted commas because it's as estimated in the corresponding meta-analysis. So if it's bigger than the expected, then it's you might say it's biased in one direction, and lower than expected, it's biased in the other. So now we have a measure by which people are getting it right or wrong. What's that related to? So one thing we thought it might be the amount of money people get. So we put it in terms of funding, and we looked at this is for countries now. We looked at the GDP of countries, divided up the research and development by the GDP, and you can see there's a nice linear relationship. So the more money a country puts into research, the more likely it is that its scientists will find significant findings. Yes, so not necessarily true, it's just they're finding significant stuff. So that's one answer. And another aspect is to think about the journal they publish in. So the journals are graded by their impact factor. If you have a high impact factor, you're more likely to be sort of an influential journal. And the journals that we're interested in publishing in Science and Nature and so on have very high impact factors. So this is going to be my last data slide for you. This is the impact factor on this scale. And then this is our bias index on this axis here. And you can see that there is a sort of linear relationship, it's significant, between the impact factor and the bias index, telling you that journals which have a higher impact factor are more likely to publish biased results. But there's another feature from this graph. So we indicate the size of the study by the size of the ball. And remember, a very big study is better powered and more likely to give you an accurate result. So now look at the relationship between the size of the study, the more accurate study, and the impact factor. So the bigger studies are down here. There are only a small number of journals with a very big impact factor. You've been seeing many of those up to now, publications I've shown you, Nature and Science and so on. And it's, rather surprising, they all have very small circles. So large impact factor, more biased, less likely to be correct, which would indicate that if you really want to find a true result, you should probably be reading a journal that no one's ever heard of, because they're more likely to be telling you the truth. And this is just some reading for you to take home. I'd strongly recommend that you read this. It's all freely available, and the title says it all: Why Most Published Research Findings Are False. Okay, that's the end of my talk. You're free to ask me questions. So since we've been doing this sort of work, that is indeed, there are now requirements from journals that when you submit papers, you should tell them what your power is. But there are many ways to write a power calculation, and any statistician will tell you, you can always fiddle it to make it look good. So how you get over that problem is a little bit more difficult. The critical thing here, and the reason that it's quite easy to get away with this, is because in many cases we don't know what we're trying to detect. So perhaps the most useful thing I showed you was the graph of the effect sizes in genetic studies, because genetic studies have now got over some of these problems, and the estimates we get are now pretty reliable. So you should be able to power a genetic study very accurately. But science is doing things that no one else has done before. You're trying to make advances into the unknown. So if you're doing a study and you're trying to find something which you don't know whether it's there or not, you don't know before you do that study what the effect is. So you don't really know how to power it. You can say, look, on the basis of what other people have done, I think it's like this, but you may be wrong.
So how, with like the higher impact factor journals, they have smaller studies, are they still able to get published in like these famous journals?
They did.
Yeah, well, just because it's like a shocking result, or are they like?
So if you're an editor working in one of these journals, you're getting in papers coming in like piles and piles, and you're looking at them, you're making snap decisions. Oh, I'm not going to send this out to review, just send that straight back, and this one maybe. And the scientists are all there thinking, my God, my tenure's coming up. I haven't got a paper in one of these big journals. I've really got to convince the journal to publish it. So there's a little sort of war going on here. And the scientists are writing stuff to really sort of make what they look like as being important and novel and catch the eye of the editor. And the editors are sort of trying to fight off this flood of stuff that's coming in. So the bias is a technical thing here, in the sense that what we've done is we say, look, we don't know, we don't actually care what the right answer is, but we've got maybe 50 studies that have been carried out, all on the same subject. So they could all be testing the relationship between the serotonin transporter and neuroticism. We have those 50 studies. So we can combine all of those, and I showed you a graph at the beginning of a meta-analysis where you saw each study has an estimate of the effect size, and at the end we pool those and end up with an estimate. Now we don't know whether that's correct or not, but we take that as the truth. And now we say, okay, so overall everyone's estimate is 5 or whatever it should be. And then we go back to the individual studies and say, hey, you made it 10, you're overestimating, but you guys made it -6, so you're underestimating. So some people are biased one way, some people are biased the other way. That's all it is.
Like that bias includes both ways?
Of course, yes, over and under, yeah. And the interesting thing is that overestimates tend to be the ones that you see. And then there's a whole other argument about actually many people, if they don't see something, won't publish it. So we know that there's going to be bias in the literature because many people will publish positive results. Others will do the same experiment, not find it, and then not publish it.
Are resources one of the, like financial resources, the main constraint when you're powering a study? Are people less likely to make a well-powered study?
Because it costs more? It's a good question. I mean, in theory, not. I mean, you're supposed to power your study by, but I'm sure that must be, you know, if you, there's a number of ways of thinking about this. So if you know that you've got a very small effect size that you're supposed to be seeing, and then you work out you need to study 1 million people or whatever it is, and then that means you've got to have a grant that is costing $100 million. No one's going to fund that. So, well, maybe if we cut it down to only 1,000 people, you know. So there might be some things like that going on. I don't know. It's a good question.
Are there any journals or is there a movement to publish replication studies?
So there's I didn't mention this, but there's a paper in Science about 3 or 4 years ago called the Reproducibility Project. And a lot of this has come out of psychology, which is why I started this talk in psychology. And psychology has had also some of the worst problems in this field. And the Reproducibility Project recruited psychologists across the world and said, here's a paper with a result. I want you to replicate it. Here's the methodology. Please just go into your laboratory and try and reproduce it. And that was done for, I think, maybe 100 or so studies. And the results showed more or less what I showed you here, that the power was in general very low, and it didn't really work out so well for the psychologist. And this has then led to a general idea that we should be thinking more closely about our power, but we've already heard how difficult that can be. But putting into our studies much better measures of our reproducibility. So the whole idea of reproducibility has become sort of much more part of the scientific vocabulary. And now, as I've just seen, if you submit a grant to NIH, there's a whole section called reproducibility and reliability, in which you are asked to justify that you're going to meet the accepted criteria. Yeah.
If you're going to reproduce a study, like, is that like, does that mean that the people who like carried out the initial experiment messed with the results?
Do you mean fraud? Oh dear, now we're getting to dangerous territory.
Like, is that just like a normal thing for you to like?
Normal to be fraud?
No,
No.
not that. Is it like, so for example, if someone like reproduces an experiment done in a beaver and they don't get the same results,
Yeah.
and then like why does that happen, I guess?
Well, this is what my last part of the talk was trying to address. Why, you know, and I'm thinking there are probably social effects. People need to. There's a big pressure now to publish findings, and people's salaries depend on it. So it's like, you know, if you don't get your grant, you're not going to get your salary, and in order to get the grant, you've got to have a positive finding in the big journals. So there's going to be lots of pressures on scientists to do that, and we don't. You know, doing science which is robust and gets answers, but is not very interesting or doesn't really break some new barrier. It's not so, like, why would you want to do that? And so there's sort of social issues around this. Yeah. Okay.
Can you talk about how was it the research in North America or the better funded research?
So all we see is this observation that in countries where more money goes into research, then they're more likely to publish positive findings. You might think it would go the other way around. You might think that the countries which have less money are so desperate to get stuff, but it seems to, so it's not immediately obvious why that's the case. I mean, that's just the observation we made.
Yeah.
But you know, you could go and try and replicate our result.
Yeah.
So one of the interesting points that John Ioannidis makes is if you look at the hottest areas of research are the most likely to be wrong, for precisely that reason. Yeah.
Do you think in those areas, let's say, doing like registration for IDs, like pre-registering IDs?
Yes, that's a good point. So that those of you who don't know, one way of dealing with this problem is that you should register your study design before you do the study. Put it up on the internet somewhere. So this is what I'm going to do. So there can be no wiggling when you get to finally looking at the results. And this has been a problem. So for example, this is something, again, that my colleague Marcus has done, that we wrote round to all of the people who've done the interaction study, the serotonin and transporter and environment study, wrote round to all of those groups and said to them, we're going to do the definitive experiment. So everyone needs to explain exactly what they're going to do. We're going to have a completely open protocol, and everyone's going to agree to this. And what happened is, as Marcus, who, if you ever see him, you can hear from him, is that the groups who published the positive findings were less likely to take part in that study. But we weren't allowed to publish that, so I can't tell you that. Yeah.
So what are your thoughts on the serotonin moderation?
Well, I've never seen any convincing evidence for it.
Yeah.
But,
Did I see it in the Manhattan part, the GWAS, that it was 10%?
no, there's nothing. If you look, it's completely flat. Yeah. But the slight problem with that argument is that the GWAS is based on SNP data, and this is a length polymorphism. So you have to use a surrogate, and because there's always some excuse why you didn't see it. But, you know. Okay. Thank you.